week. Today we are going to look at viruses in terms of how they are acquired from an infected person to another, and once they get into the body, how do they reproduce, and then finally look at the pathogenesis, which is how do viruses actually end up causing disease in us. So we are going to look at transmission, replication, and pathogenesis of viruses in general. So the first slide, which I am sharing with you, first of all, I should not probably be sharing that slide. So when you talk about transmission of viruses, can I see, I'm sure this is actually not new. Most of you actually know about how viruses are transmitted. Can I see from the chart the modes of transmission of viruses that you know? I want to see from the chart sexually transmitted, excellent. Another route of transmission, inhalation, coughing, in airborne, body fluids, airborne, vectors, fecal, oral, contact, fetal. Hey, how is that done by fetal? Okay, that's probably mother to child transmission. Blood transfusion, saliva, and oral. So basically, very good, thank you. I think we guys actually know about the routes of transmission of viruses. So we're just going to expound a little bit about them, but the good things that you know all the route of transmission of viruses. And so so on this slide, I'm showing a pictorial representation of the mode of transmission of viruses. So the first route is aerosol through air and saliva. And so this is mainly a route of transmission of viruses which colonizes the respiratory tract and the lungs. And when they infect these organs, they cause inflammation, which causes irritation, and then leads to coughing and sneezing. So when you cough and sneeze, you are likely to transmit that virus from you as an infected person to the next one. So viruses which are transmitted via aerosol, so one way of acquiring such viruses is by inhaling. Which other routes would one then acquire these viruses other than inhalation? So we're talking about viruses which are spread through sneezing and coughing. So here I have a viral disease, I'm sneezing and coughing. I can spread it to other people by that other person inhaling the droplets. But there are also other routes of there are also other ways other people can acquire the virus. Which are these other ways? Can I see from the charts? Through saliva, so through the eye, so okay, surface contact, good. Oral, I don't know what that means, but maybe when you have contact with someone through maybe kissing, saliva exchange, that's correct. Interacting with contaminated surfaces, that is good. So remember COVID-19 virus. Thank you very much, everyone, for skin piercing. that's a little bit interesting. So COVID-19 virus was spread through aerosol, and remember we were told to wear a mask, hand washing. One of the most effective route of transmission of viruses which are transmitted via aerosol is through contact. People like sneezing on their hands, or when they sneeze, they close their mouth with their hands, and by doing that, you are spreading the viruses on your hand. And if you shake hands of someone who is uninfected, that person can be infected because if they don't wash their hands. The other route of transmission is fecal-oral. Fecal-oral is where you eat feces or murphy or stool, but do we really eat feces? Do you think we eat feces really? Is this a common mode of transmission of viruses? Yes. Can I see through the chat? Is this a common mode of transmission of viruses? No, it is not. Yes. No. Yes. So which one is which? Can you give me an example of a virus which is transmitted through fecal oral? Then we can say whether it is common or not. An example of virus that is transmitted through fecal oral: hepatitis A, correct. Typhoid is not a viral disease; that's a bacterial. So please know that. Enteroviruses, correct. Rotavirus, yeah. Polio, yes. Polio is not common because of vaccination. How about rotavirus? Is rotavirus a common disease? Is rotavirus a common disease? No, hantavirus is transmitted through fecal oral feces of rats, probably to humans, but not from human to human. So how about is rotavirus a common disease? Yes or no? Rotavirus. Is it yes? Is it? Do you know that? Are you aware of someone who has suffered from rotavirus disease? Yes, I can see people are saying yes, right? So then, if we say that fecal oral is not a common mode of transmission of viruses, then why are we saying that rotavirus is a common disease? The thing is that fecal oral, as I said, involves ingesting either a virus in form of when it is contaminated in a food or water, or fruit. So yeah, if you have a vegetable that is contaminated with a virus, then and you eat it, you can get infected. So which other viruses? Okay, so we know that fecal oral is one mode of transmission of viruses. Most viruses which are spread through this route are very resistant to environmental conditions. We saw that on Wednesday's lecture. The other route of transmission is sexual transmission, and we know very many viruses are transmitted through sexual intercourse. Which ones are these viruses? Can I see through the chat? Herpes, okay. HIV, hepatitis. Which hepatitis? Which hepatitis? Because we know HIV for sure. HPV, human papilloma virus, correct. Hepatitis B, correct. Chlamydia is not a virus. Chlamydia, syphilis are not viruses. So we are talking about viruses. So your papilloma virus, correct. 8 herpes viruses. We have 8 herpes viruses. Only 2 of them are transmitted through sexual intercourse. I'm seeing someone here called Nayo Nisup. I'm not sure how that person's name is on how do you put such a name? Because we need also to know you. Okay, so Christian, why are you? Are you laughing or you are crying? I'm seeing some images of people who are like laughing. Okay, good. You are laughing. It means that's good enough. So which herpes viruses do you think are transmitted through sexual intercourse? We'll talk about these viruses in future, but I wanted to know because these are common diseases. So probably we should be knowing about them. Do you know the herpes viruses that are sexually transmitted? Yes, can I see on the chart? Herpes simplex type 2, correct. And which other herpes virus has potential for sexual transmission? Type 1, correct. So you guys are very learned. So that's good. So you've mentioned all the viruses that are transmitted through sexual intercourse. And now we move on to viruses which are transmitted by vector. We call them arboviruses, mosquito-borne viruses. So vectors can transmit viruses. Example of viruses which are transmitted by vectors, I want to see on the chart: dengue fever virus, Zika virus, yellow fever virus. Which other viruses? Which other viruses are transmitted by vector? Zika, yes. People have said Rift Valley fever virus, chick. There's a no virus called chick unless you're referring to chikungunya. West Nile virus is transmitted through the correct typhus. Oh my goodness, arbovirus, Bunyaviridae, good. So a number of viruses, West Nile, good, are transmitted through by mosquitoes, ticks, and these viruses, this mode of transmission is called vector-borne, arthropod-borne mode of transmission. And then we have transmission by, dogs or other animals, like, dogs can transmit viruses as well through dog bite. Other animals like fleas and rats can also transmit viruses. In addition to this, we also know that there is mother-to-child transmission of viruses. Mother-to-child transmission of viruses. So mothers can also transmit viruses, either through breast milk or when they are delivering the babies. So these are the main mode of transmission of viruses. So you should be aware that viruses are, whereas they are very small, they are very smart. They have targeted different routes through which they can enter, our bodies, and as such, we can't avoid having viruses in our bodies. So a little bit more about transmission. So we've just talked about routes of transmission. That is how, how do someone acquire virus from an infected person? But looking deep, this acquisition, at what point in your body does it occur? I want us to take a look at what we call port of entry for viruses. And I want to start with respiratory tract. Respiratory tract, this is usually the easiest, we call it the easiest port of entry for most viruses. The reason is because we eat things through our mouth and they end up with we eat and inhale a lot of things from the environment and they end up within our respiratory tract. And the respiratory tract is also covered by epithelial cells and mucus. Mucus is like it's like a treadmill for viruses. It takes them down to the lungs as well as to the lower part of it. Viral diseases, according to different groups, these are the most common viral diseases. And the examples are on the. I've put up the examples on the slide: mumps, measles, Coxsackie, rhinoviruses, coronaviruses. All these can actually get through the respiratory tract. The other port of entry of viruses is the gastrointestinal. So the viruses which get through our mouth, but they end up in the stomach and the small and the large intestine, and where they actually cause disease. They infect the cells that are lining these organs and get entry into our body. Some viruses use this port of entry as a passage. They infect the stomach, the small intestine, and they get into bloodstream through that and move to other organs which are forming the target organ. So the other route of port of entry is genitourinary tract. This is these are the viruses that are transmitted through sexual intercourse, but we know the vagina, the urinary tract, are also port of entry of viruses in our body. But these viruses, they have to be able to break, the epithelial cells that are lining the urinary tract rather, and the vagina to be able to establish infection. And the examples of viruses that do this have been listed on this slide: HIV-1, 2, hepatitis B, C, herpes type 2, and papilloma viruses. We have actually seen this earlier. Skin is also another port of entry into our body. However, our skin, to a large extent, protect us from viruses because the outer part of the skin is usually composed of dead cells. And the dead cells, usually, viruses don't infect dead cells. So we have what is called innate immunity against viruses, and skin actually form part of that. So should a virus land on your skin, the virus has no opportunity to infect you unless the layer that is composed of dead skin is broken. It is only when it is broken that the virus gets into your body. So skin is another port of entry, but that only happens when the dead layer of the skin is broken. So I want us to move to the next subtopic of this lecture, which is viral replication. And in our Wednesday lecture, I mentioned to you that viruses are really not living. They're not living, but they perpetuate themselves in nature. They are able to reproduce. They are able to make their baby, viruses. But they do this using the living cells of either humans or other living things. And so in today's lecture, we want to understand how do these non-living entities able to make new copies of themselves. They do this through a process called replication. And this is done through 7 or 6 stages, depending on the type of the virus. So the first step is the attachment of the virus onto a host cell. Then the second step is the penetration of the virus through the same cell. And the third step is uncoating. Uncoating only occurs for viruses which are enveloped. So if you have an envelope virus, then when they are trying to penetrate the host cell membrane, they leave their coat and they then release the nucleocapsid of the virus into the cytoplasm of the host cell. Then the next step is transcription, which we are going to talk about this slightly in the next slide. We have transcription, translation, assembly, and release. So if you think about bacteria, I don't know whether you have learned anything about bacteria. Bacteria reproduce through binary fusion. So one bacteria divides into 2, those 2 divide into 4, and that's how they continue. They continue making themselves. For viruses, it is a 7-step process. So, and this 7-step process must involve a living cell because they lack a lot of things for them to reproduce. And so let us look at these stages. So I talked about the first stage, which is the attachment. So at this stage, remember when I was showing you the shape and the structure of a virus, the envelope viruses, I showed you something like a club, that hangs on the envelope of the virus. That component of the virus is what the virus used to attach onto a host cell. That is the first interaction between a virus and a host cell. as it plans to reproduce. That club-like structure on the virus, it must look for a specific structure on the host cell. And this specific structure on the host cell is called a receptor. So the interaction between a viral ligand, which is what looks like the club-like structure on the virus, the interaction between a virus ligand and the receptor on the host cell is like a key and lock kind of a thing. It is so specific that a virus will not attach to any surface of the cell and initiate attachment. It must find what we are referring as a receptor to attach to. So is the word receptor familiar to you? Have you ever heard the word receptor? Can I see on the chat? Have you ever heard of the word receptor? If you have, can I get an example of what a receptor is? Yes. So can I get an example from you guys? Give me one example of a receptor to a virus. CD4, correct. So you guys have heard. So CD4 is a receptor of HIV virus. Thank you very much. At least you've known about this. So if you think about CD4 and HIV, CD4 receptor is only found on T cells, which are called CD4 positive T cells. And these cells are found in blood. So when you acquire HIV will not infect B cells. HIV cannot infect epithelial cells. It only infects T cells, which are expressing the receptor called CD4. So what I'm trying to say is the attachment of a virus to a host cell is so specific that if a cell is missing the required receptor, it cannot attach to, the virus cannot attach to that cell. But if the receptor is available, the virus will attach to the receptor, and then the next thing it does is to try to penetrate. Now, penetrate means the virus is trying to pass through the cellular membrane. And by doing this, it depends on now whether the virus is enveloped or the virus is naked. So for envelope viruses, the viruses leave the envelope outside on the membrane and then releases the genetic material, the DNA or the RNA, into the cytoplasm of the host cell. For the naked viruses, they penetrate the host cell membrane by what is called translocation or endocytosis of the membrane of the cell. So because they don't have the membrane, they don't have the envelope, they push the cellular membrane. Is like, you know, you're trying to push a football, you know, the football, the way it is, until you're trying to push a metal until, you know, the surface of the football gives in and allows it to get inside. So that is how the non-envelope viruses gain entry, a process called penetration. Now, the third step and the fourth stages are called transcription and translation. Transcription, so what happens once the genetic material of the virus has been released after penetration? The genetic material is usually released in the cytoplasm of the host cell, and it could be either an RNA or a DNA. We know that viruses have either DNA or RNA. And so, once it releases the genetic material in the cytoplasm of the host cell, the first thing that happens is that the virus must make a messenger RNA. We talked about messenger RNA when we are talking about the Baltimore classification. The virus, the DNA or the RNA of the virus is used to make messenger RNA. Why do they need to make messenger RNA? Because we remember we are talking about replication, producing a new virion. And viruses are composed of proteins, so they have to synthesize protein. The only way they can synthesize protein is by generating a messenger RNA. Now, the process of producing messenger RNA, either from a DNA of the virus or from the RNA of the virus, is called transcription. That process is called transcription, formation of messenger RNA. Now, I want you to remember two things about transcription. We said earlier that we either have DNA or RNA viruses. For RNA viruses, the transcription takes place in the cytoplasm of the host cell. For RNA viruses, transcription takes place in the cytoplasm of the host cell. For DNA viruses, transcription takes place in the nucleus of the host cell. For DNA viruses, transcription takes place in the nucleus of the host cell. Now, when the messenger RNA is formed, whether it is in the cytoplasm or in the nucleus, it is usually released in the cytoplasm, so that it then start interacting with ribosomes. We talked about ribosomes on Wednesday, so I still hope that you remember what ribosomes are. Now, the messenger RNA, the process of messenger RNA interacting with ribosome is basically to translate the genetic code in the messenger RNA to produce viral proteins. And this process is referred to as translation. The process of this process is referred to translation, the formation of viral proteins. So how this happens is that, so a ribosome will interact with the messenger RNA, and it starts, as it reads the codons, it produces peptides, and these peptides, they become a chain and releases them. When they become heavy, the ribosome releases them. What that means is that different parts of a virus are produced in bits. They are produced in bits. I'm imagining. Let me give use example of a human being, for example. You can imagine if human beings were viruses, when fertilization takes place, the head of the baby, instead of being one, you end up with several of them in the womb of a mother. That is how different parts of the viruses are made during the translation. So you have so many heads, you have so many hands, you have so many legs. All of them are, found within the cytoplasm of the host cell. I can see some people are sharing some notes on the WhatsApp on the chats. Sasa nyamaza tusikie mwenye analipwa. I don't know whether I'm the one mimi ndio mwenye analipwa ama ni mtu mwingine. The different parts of the viruses are made. So like the capsid, we talked about the capsid as a component of viruses. So you will find several, proteins consisting of capsid are found within the cytoplasm. If proteins related to the envelope, not necessarily envelope, but some viruses do carry enzymes. These enzymes are also found in bits within the cytoplasm. So once this translation has occurred, there are enzymes that will do what is called assembly. They will go and start looking for the head connected to the neck, to the thorax. I'm just using those words for you to understand. the legs, the hands, and then you have a complete virion. So that process is called assembly. So the viruses are assembled in the cytoplasm, and then once they have been assembled, they then move to the membrane of the host cell, and then they are released. So what I've been talking about, I know I've been talking about a lot, but I want you to look at this photo. You're able to see my cursor, right? Are you able to see my cursor? Anybody? Yes. So this is a host cell, right? And with a nucleus. This is a virus. We're using influenza as an example. So the first step is the attachment. So the influenza virus, the spike protein must find a receptor to bind to. You see this virus here, it has the genetic material inside, so it attaches to the host cell. Then the next step, it undergoes penetration. It penetrates until it releases the genetic material only. Like for influenza virus, it's able to push also the envelope with it through the membrane. Most viruses would leave the envelope on the membrane of the host cell. So for the influenza virus, it pushes the envelope, and then the envelope dissolves, and then it releases the genetic materials. You see the genetic materials now are released within the cytoplasm of the cell. So when that happens, then you go through what is called biosynthesis. Biosynthesis mainly is what we are referring as transcription and translation. This is formation of messenger RNA and formation of various viral proteins. And you can see there are many, they are littered all over within the cytoplasm. And then you start with assembly. So you then form single virions, one by one. You can see them. And then when they are formed, they then move to the envelope of the membrane of the host cell, and then they are released. So this is what happens in the process of the replication of viruses. Now, at the stage of release, when these viruses are released, it also happens depending on whether the virus is an envelope virus or whether it is a naked virus. For envelope viruses, they leave the cell through a process called budding. Budding, B-U-D-D-I-N-G. So for envelope viruses, they leave the host cell through the process called budding. What that means is that you see this virus here, it comes and starts pushing the membrane. So the membrane swells, and it keeps on pushing it until it takes parts of the membrane of the host cell. And remember, on Wednesday, I was explaining to you that the envelope of viruses consists of viral protein as well as the host cell membrane protein. So for envelope protein, they take part of the host cell membrane, and then they live with it. Then they repair, that cell that they repair that area, and the cells still remain alive. For naked viruses, so envelope viruses are very friendly. They most of the time they don't kill the cells, but for naked viruses, they're not very friendly to the host cell. Once they have been formed, like at this stage, when they come to the membrane, instead of pushing the membrane so that they can take part of it and repair like envelope viruses, they make an incision, okay? A process called lysis. They cut the membrane of the cell to allow them space to live. When they cut the membrane of the cell dies, and then they release themselves. Remember, when viruses are produced within the cell, they don't just produce one virus. They produce thousands and thousands of viruses, which then they release into the neighboring environment. Okay? And so I want you to take a look at this photo, and maybe is there something that you can point that is different between these two photos? Is there anything that you can look at and tell me the difference? What is the difference between these two photos? Receptor. What else? Transcription and translation. One virus is naked, the other one is envelope. Site of transcription, correct. One is in the cytoplasm, the other is the nucleus. Very right. That's very correct. So that is what I was looking for. And this is what I was trying to tell you, that for RNA viruses, the transcription is taking place in the cytoplasm. For DNA viruses, it is taking place in the nucleus. And so that is basically how viruses replicate. And the next thing we want to look at is the spread of the virus. So once the viruses have replicated, they have produced thousands and thousands of viruses in your body, then they have to look for a new host. And this will be done through the transmission that we have talked about. So a virus is spread now to the other host. And when it enters to the new host, the first thing that happens is that the virus can replicate and remain at the site of entry, or the virus can move to the target organ, which could be far away from the point of entry. So the spread of viruses can be looked at, through three different ways. The first way is what we are referring to as localized infection. And localized infection is where a virus enters into your body, either through the mouth, the skin, and then it replicates only within that locality, and does not spread to other parts of the body. Example of these viruses are like the rotaviruses, the viruses that cause that cause gastrointestinal infection. They end up causing just local infection. Some respiratory viruses also end up causing infection within the respiratory tract, and they don't spread beyond the respiratory tract. Other viruses have developed an approach where their spread is called systemic infection. Systemic infection means they enter into your body through a port of entry, like what I mentioned to you earlier. And then, for example, the mouth or the skin, the first thing they do is to replicate at the point of entry. And thereafter, then they actually spread. They get into blood and spread to their target organs. So you find, for example, a virus like polio, you will ingest it. You will ingest it through the mouth. It will go into the gut. It then, replicates in the gut, produces a few copies of poliovirus. Some of it is released through feces. Others get into the blood system, and then it finds its way into the central nervous system. So those viruses have developed an approach of spread called systemic infection. For viruses that uses the blood to reach their target organ, the process is called viremia. So once the virus is in blood, we say, oh, so-and-so is viremic. It means he has virus in blood. Viruses have also adapted. Some viruses are spread, not through blood per se, but through the nerves, the nervous system. So, for example, when you get bitten by a rabid dog, the rabies virus does not get into blood. It moves through the peripheral nerve system to reach, your central nervous system, and that is where it causes damage. So this is common with viruses like rabies. We know of other viruses like herpes viruses also. Some of them prefer using the nerve system to spread within the body. So finally, let us look at pathogenesis, because I'm cautious about our Muslim brothers and sisters. I want us to finish this within the next 5 minutes. So let's look at pathogenesis. Today, we started by looking at how viruses are transmitted. We have seen different modes of transmission of viruses. We have also looked at, once you acquire the viruses, how do they replicate? How do they reproduce their own variants within your body? We also look at the spread. Once they get into your body, then how do they move from the port of entry to either the target organ, where they now, replicate more and then cause a disease or not. So the final thing we want to look at is pathogenesis. The word pathogenesis, if you look at it, it mainly referred to the outcome is a disease kind of. We talk about pathology, the damage caused by, a virus or a bacteria. But I want to remind you that most viruses that infect human beings have no intention of causing disease in humans. Neither do they have intention of killing humans. Causing disease and killing human beings is an accidental outcome of viral infections. Most viruses would want to get into your body, replicate without causing disease, but live with you forever and ever. And for your information, all of you in this class, you are carrying a number of viruses. You may not be sick, but if you were to take blood from you today, I would promise you that we'll find 1 or 2 viruses. And these viruses are very friendly to our body. However, the infection of certain viruses actually lead to disease. And how this happens is what I want to explain to you under this subtopic of viral pathogenesis. Now, when you acquire a virus, through any of the mode of transmission that we talked about, and I've just explained to you that, the port of entry of a virus, whether it's a skin, gastrointestinal tract, respiratory tract, when the virus gets into that first port of entry, the virus would replicate, to make sure that it has some sort of footing in your body. If that is where it wants to cause disease or wants to stay, it will replicate extensively, or it will replicate and hide in that region. Other viruses would actually move on to other organs. But as soon as a virus gets into your body, your immune system also recognizes that there's a foreign body here. And what does your immune system does? Your immune system would want to clear the virus. Okay. So you guys must have done immunology, I believe. What are the 2 levels of immunity that our body, uses to clear infections? What are the 2 levels of immunity our body uses to clear immunity? So the body uses innate immunity. We also use acquired immunity. Innate immunity are, things like the skin, the tears, the mucus, those are the first line of defense. So imagine a virus like, COVID-19, which now everyone knows about. You have inhaled it. The first thing that would happen is that it gets into your throat. The mucous membrane is there. The mucous membrane does not allow it to get into the epithelial cells, which it may infect. And the mucous membrane can actually push it down to the stomach, which is acidic, where it dies. So that innate immunity alone is able to clear the virus. So if by chance the innate immunity is not working, then, and this can happen as a result of, say, you have a broken skin, then the virus gets entry into your body, then the adaptive immunity now comes in to try to clear the virus. So what happens is that every time a virus gets into your body, your immunity is there, ready to fight it. Most of the time, our bodies are able to fight the new entry. A lot of times, in certain cases, the virus actually overcome our immunity. And once they overcome our immunity, that is when the virus now spreads and it is able to cause disease. So pathogenesis is about the interplay between the virus trying to establish itself and your immunity trying to fight it. If the virus wins, then the virus has an upper hand of replicating in the target cells and causing disease. Okay? So finally, I always like using this slide to explain pathogenesis, at the cellular level, because viruses target cells. I want us to start with where my cursor is. We have this virus here. It's a black dot. This is your cell. It could be any cell in your body. So we said that when a virus, when you get infected, the virus will attach to this cell. And the interaction between this virus and this cell, there are several outcomes. There are several possible outcomes, right? And those outcomes can be explained after this stage of infection. So number one is that, let's take this arrow going up here. This virus that has already gotten into the host cell, remember the virus would replicate, right? Some viruses have got the ability to cause, to transform a cell from being a normal cell to become cancerous cells. Some viruses have the ability to transform a cell from being a normal cell to become cancerous. So how does this happen? So all cells in our body have a lifespan. They live for a certain number of days or months, depending on the type of a cell, just like the way as human beings also have a lifespan. And what happens, for example, if a cell has a lifespan of 20 days, so by the 15th day, internally within the cell start getting signals to start dying, what we call programmed cell death or apoptosis. By 18th or 19th day, internally the cell dies because of the programmed cell death. That happens with majority of the cells in our body. And so what happens is that certain viruses, when they get into your body, they have the ability to circumvent, to stop those signals from kicking in, so that instead of this cell dying after 20 days, the cell now acquires a different characteristic, whereby instead of dying, then it starts dividing into two cells. So you have one cell dividing into two cells. These two cells divide into two. Then they become 4. 4 divides into 8. Then 8 divides into 16. Then you end up with what we call cancer. Are we together? So cancer, we now know that there are several cancers which have a link to viruses. Any example of viruses, cancers that you know are linked to viruses? Any examples? Can I see on the chart? We're almost winding. This is the last slide. HPV. I'm talking about the cancer and which cancer? Cervical cancer, correct? Which other skin cancer? Which skin cancer are you talking about? Be specific. Cervical cancer, yes, cervical cancer. There are many cancers that actually have a link with viruses. But I think at this stage, Kaposi sarcoma, yes, caused by which virus? Which virus causes cancer? And Kaposi sarcoma. Liver cancer, yes, liver cancer is caused by, Kaposi sarcoma is caused by herpes virus 8, correct? Liver cancer is caused by hepatitis viruses, correct? Yes, so this is one way by which viruses cause disease in human beings. We started from here and we ended up there. The other mean by which viruses can also cause disease and the death in humans, and let us start from here. We have gone through this arrow, is when the virus, instead of transforming the cell, the virus produces several copies of itself within the cytoplasm of the host cell. And then when the virus is leaving, it cuts the membrane. Remember, we talked about the naked viruses. What they do, they cut the membrane of the host cell and kill them. Now, this process is called lytic infection. So if you have an organ, say the liver, where the liver cell, hepatocyte, get infected with, very many viruses, and they replicate, then they will kill very many of those cells. They cause inflammation, which then leads to disease. So some viruses, because of the nature by which they replicate, are able to kill the cells, and the cells are the building block of the organ, and the organ become, inflamed, and we end up sick. So that's another way by which viruses cause disease. Other viruses still follow this, the third arrow. If you look at this cell and this cell, there's a difference. This one has got a lot more viruses within the cytoplasm. This one has got very few viruses within the cytoplasm. Other viruses, so these viruses are really lethal. They cause disease nearly, in almost all the people that they infect. Other viruses adapt a more friendly approach in terms of how they live with humans, whereby they produce very few copies, just release a few of them, and then end up with what is called persistent infection. An example of a virus that causes that is hepatitis B virus. The last group of viruses are even more friendly. These viruses, they produce very few virions within the cell. They rarely release them. These virions stay within this cell, and they don't kill the cell. So you end up with what is called latent infection. And there is a family of viruses that cause latent infection. Example of the family of viruses that cause latent examples, can I see? So Ashley is asking, when the cell dies due to programmed cell death, how does the tumor comes about? So Ashley, I'm saying that when you get infected with viruses that cause cancer, they shut down that process that leads to programmed cell death. So it doesn't happen in that particular cell now. So that cell now does not die, it becomes normal. And then that's why you end up with the tumor. Tumors are growing, right? So I was asking the question of which viruses are associated with latent infection. Herpes simplex viruses are associated with latent infection, chickenpox virus, cytomegalovirus, EBV, very good, William, and the rest. So this is how viruses, varicella zoster virus is correct. This is how viruses cause disease in humans. And because of time, I hope we have covered this lecture very well. And I want us to end there so that we can let our brothers and sisters to go to mosque and pray for us. Thank you very much and enjoy the rest of the afternoon. Bye-bye.